Welcome everyone to our second session on clinical data and how machine learning can empower clinicians to use and amend data properly. Today we have a number of very interesting and exciting expert guests that Andrew will lead a conversation with about what we've talked last time, and certainly a few more topics will crop up. First of all, just a reminder that we have two more sessions in the first half of the year before we schedule the second half. One is on the 17th of May and one on the 15th of June. So already put those into your diary. Before we start with our session, I'm very happy to introduce our next CCA summer school that will come up in September. It's for students, researchers, clinicians, and industry. So it's very consciously designed as a community project of all sides of the field. It will happen from the 11th to 15th of September, and data will be a huge topic among many others. AI, machine learning, very practical approaches, very hand-on approaches in some of our seminars. So please go to the CCAM website, look for the summer school, and hopefully we will see you there. Now, I will hand over to Andrew and our guests, and I'm very excited to hear what they will be talking about. Andrew, the floor is yours.
Thanks, Andreas. Welcome, everybody. I'm going to introduce our panel a little bit in a few minutes' time, because today is a very important topic, which is sort of the Cinderella, if you like, of artificial intelligence, which is the data that AI so much depends upon, and it's usually doesn't go to the ball often. And our goal today is to, not only have it go to the ball, but get its slipper and get its true place in the hierarchy of importance in the AI world. And there is no one who has pioneered this more than Professor Mihaela van der Schaar, obviously the founder of the Schaar Lab and of the Cambridge Medical AI Center as well. And we wanted to start by Mihaela recapping on the key challenges for data in AI and on the main techniques that can be used, many of them pioneered by Mihaela's lab, for dealing with those challenges. So I'm going to hand over to Mihaela. She's going to speak for a few minutes, and then we have a wonderful panel that have been using these and other techniques in the wild, and we're going to have a discussion with them, and that will be our opportunity also to ask your questions from the audience, because what I'd really like is to have the conversation with all of you as well as with the panel. So we're going to switch our cameras off and hand over to Mihaela to give us the recap of the challenges and the techniques for data in AI in medicine.
Thank you very much to everyone for coming and joining us today. I'm going to make a very brief recap of last time when I introduced 4 machine learning antidotes for improving clinical data. The challenges associated with clinical data are numerous. And when I talk about clinical data, I think here about electronic health records, clinical registries, biobanks, and much more. And we know that the performance and robustness of any type of analytics, machine learning included, depends on the characteristics and quality of such data. And the imperfections of clinical data abound. And partially this is because this is very valuable and complex data. In some settings, we have imperfect data quality, noisy data, corrupted information, ambiguous information. We may have biased data. We may have missing data, and this data may be at times missing because it's unimportant. At other times, may be missing at random. We also have in many clinical settings limited data. That was definitely the case in COVID, when initially in the pandemic, we had only very limited data to learn from and make decisions in. And in other settings, like rare diseases, this remains a challenge. And I'd like to highlight the fact that these challenges are important for machine learning, but they are more generally important. And what's interesting is that, as Andrew said, usually we think about machine learning as being an analytical tool that ingests data and issues predictions, causal inference, etc. Here we are using machine learning to do something very different. We use machine learning and machine learning techniques to improve the quality of data. So the focus is not on building analytics. In fact, you can use statistical methods, epidemiological methods after you have cleaned and improved your data using machine learning. And the 4 antidotes were data-centric AI, synthetic data, self and semi-supervised learning, and combining knowledge that clinicians may have or pharmacologists may have with AI and machine learning models to bootstrap the process when we have very limited data. So we, again, have this new lens for machine learning, which is not focusing on machine learning for analytics, but rather machine learning to improve the quality of the data. And what we see is that machine learning can help with pre-processing and clean data. It enables you to do data curation, cohort creation, data quality evaluation, and maybe even improve and augment your current data. We also use this data-centric lens to improve the type of models that you can choose for prediction, and also what type of data should be used and how should the data be used to test any type of hypothesis or analytic that you build. And finally, at deployment time, we all know that when a particular methodology or analytic has been used in practice, many things change. Many things change over time, but also many things change because of the involvement now of the clinician and the patient with these tools. So as the data is changing, is shifting and drifting, or as a particular cohort is changing, or as the disease, like in case of COVID, changes, we may need to develop new technologies using data-centric AI that enables us to really, without retraining or with minimum retraining, be capable to continue to use the analytics of interest. You can read about this on our website, where we put together not only a thought piece, but also a collection of hands-on tools that you can use to really bring this data-centric lens to improving the quality of your data at these different stages. Another one is synthetic data. We talked quite a lot about synthetic data in the context of revolutionizing healthcare. And the focus here is not only to create synthetic data for exchanging it with others because of privacy considerations. That was the initial use of synthetic data. But increasingly, we think about creating better data, so synthetic data being better than real data, and partially because it can create versions of the data that remove biases, that are able to augment in small sample size cases the data sets, that can enable us to transfer data from one particular hospital or country to another, and finally roll on future scenarios such that you can do future planning. So create data that has never been seen before. This would be especially useful, for instance, again, in COVID, where one could see the pandemic evolving in different types of way and create plausible scenarios for that. So the data is not even existing, but we can extrapolate credibly from the current data. The third one is the notion of self-supervised learning. And the idea here is that we are trying to learn not only from data that is labeled and for which we know, for instance, the disease of interest, but we can use other sources of data that may be either directly or slightly related to the disease of interest. So such that we don't learn only from small and well-curated labeled data sets, but also we enrich these data sets with additional data sets from which we can learn related information. And this is a very important area of self-supervised learning, and it is this technology that really has empowered the success of methodologies such as ChatGPT. Finally, if we do not have enough data, another way to provide an antidote is by using your expertise, the clinicians. So use whatever knowledge you have about the importance of variables, the independence of variables, or if we have in pharmacology, you may have some information about how a particular drug will behave in the body. In this type of settings, these models themselves may not be enough, but when combined with machine learning through a process of hybridization, they can be extremely empowering and do much better than just one thing alone. And finally, this whole world of causality and causal deep learning is yet another way in which causal hypotheses that you may have, either validated or conceptual, could be put in as priors within the machine learning framework to improve the quality and enrich the quality of the data that you have.
So I'd like to do just a basic introduction from my point of view, and welcome to the panelists. And then I'm sure that they will be introducing themselves, and I'll be inviting them to introduce themselves and what they work on and their relevance to our discussion today as we go round. So today we have Professor Evis Sala of Gemelli. And in fact, we have only professors today, because in addition to Professor van der Schaar, we have Professor Sala, we have Professor Ari Ercole of Cambridge, we have Professor Martin Wagner of the hospital in University Hospital in Dresden, and we have Professor Suetonia Palmer of the University of Otago in New Zealand, and I hope we had briefly, and he disappeared, he's actually in a car. We have Professor Doug Bell, who I'm sure will join us briefly again.
Yeah, I'm sorry, I'm on the road. I can't hear and see at the same time, so I have to just hear. Sorry.
Okay, well, not only can you hear, but we can also hear you. So that's the most important thing, Doug. So, and Doug Great. Is from UCLA, so we're delighted to have such a distinguished panel with us. So, you know, the panel obviously deals with these issues on a regular basis, but I wanted to hear from each of the panel to give our audience a bit of a glimpse of your connection with machine learning in a clinical setting. So perhaps you can use this as an opportunity to tell us a little bit about yourself and about how you have used and how you do use machine learning in your work. So, Evis, can we start with you? Would you like to, explain to the audience exactly why this is, something that you have pioneered and how you use it in your work?
Let me unmute myself. Thank you for the introduction. Thank you, Mihaela, for the invitation. And it's good to see you from the other side of the, the border. So I've moved to Rome recently at Gemelli Hospital, but I think most of the experience actually so far will come from working in Cambridge. I'm a radiologist, so a lot of my experience will be on imaging, so using the machine learning on imaging and on data integration. So one of the main research, I don't think we actually have used so far machine learning in real clinical work. And I think let's just start being honest, at least in my field, and at least because this is the reality. And I would like to see that used in the clinical work very much, because there are lots of needs we have for that. So two of the areas I've used machine learning is in ovarian cancer, for example, and in the setting of prediction of tumor response to treatment in the neoadjuvant setting, and that is in the data integration domain. So not just imaging, but also integrating the data. And I've actually encountered a few problems that Mihaela absolutely fantastically and beautifully described. But I think imaging adds another component, which is the heterogeneity of the imaging data per se. So we've got the heterogeneity of the disease. In ovarian cancer, you've got 3 levels of heterogeneity, which you might not have in other areas or tumors. You've got within patient, so we've got within a lesion, in between the lesions, and in between the patients. So within patients, between patient heterogeneity. And this is something that I don't think, at least I'm not aware, that we have addressed quite fully. Second, it's the small sample size, because ovarian cancer is actually a rare disease. And I think as much as there are efforts to actually combine and use the large data sets, there are lots of impediments as well, you know, so, and I think it's time to address them. The other thing that I found difficult is the different scales, especially when you start integrating the data sources are very heterogeneous. The data scales are heterogeneous too. So, you know, imaging is obviously macroscopic in a way, what you see, and then you go through to genomics, which is really, you know, a different scale. So those are the problems, you know, that I have encountered then in that arena. In prostate cancer, which has been in a setting of a startup and, you know, taking something from science to sort of implement it. That's the work I've done with real world data in terms of 6-7 NHS sites with very different data sets, with various scanners, with various parameters, with sending pathology from electronically beautifully done to photos from, you know, the screen. So that arena, that kind of heterogeneity data, and the need to actually come with solutions. Sorry, I went for too long, but I just, you know, wanted to give a full perspective of.
No, it's great, because the reality is that the whole point is that the data is messy and difficult, and it's all about finding those techniques that, you know, you were referring to, Mihaela's referred to. So please don't worry about going into the specifics, because the specifics is precisely what we're talking about today. So I really appreciate it.
If I can add, it's actually also the unlabeled data, because it's so laborious to label images, data in imaging. So I actually, if anything like imaging, it just suffers from this unlabeled data and the worry. Actually, you start with a big data set, and you end up with a small one, because you start excluding things that you can't label them. And actually, we're throwing information, data out. Should we?
It's a really good question, and oddly enough, Mihaela was just talking about that just before this meeting started. So the question of how to avoid throwing data out is a very hot topic in the lab.
And I'll stop here.
So, Evis, thank you so much. That was a great start to our discussion. And I should say that I'm sure people have lots of questions. Do put the questions in the chat on Zoom, and I'll monitor those. And as soon as we've built up a little bit of a flow and momentum in the questions, we'll swap to hearing from our audience and their comments and questions. Ari, can we turn to you now? I think your experiences come from a different perspective. So let's hear from you, your connection to, you know, the data issues of AI.
Yeah, no, absolutely. So I actually started my research career before medicine in experimental physics, and I did a lot of signal processing and electronics and so on. I moved over to medicine. I'm actually a consultant in intensive care medicine. I specialize in neurointensive care, neurosciences intensive care. And quite naturally also got into data science within medicine as well. It was part of the plan, really. And you might not, well, intensive care has been in the press with COVID quite a lot recently, but you'd be forgiven for not knowing too much about intensive care. And generally, I'd strongly recommend as a lifestyle choice not having too much to do with people like me, because it means you're really, really sick. And actually, intensive care episodes are by far and away the most data-dense patient episodes that you'll see in a typical hospital. And they're data dense in a really heterogeneous kind of way. So first, we do an awful lot of laboratory tests, an awful lot of physiological monitoring. And that physiological monitoring may range from minute by minute. The tests may be day to day or every few hours. It may be irregularly sampled. some of our physiological monitoring, and a lot of my work has been working in sort of waveform analysis and trying to make inferences about physiology. Some of that is digitized to 250, 500 hertz. So, we're looking at gigabytes worth of waveform level data, and it also has inaccuracies, it's got missing data, it's got noise and various things that we've actually used in a research setting, at least AI techniques and self-supervised or semi-supervised learning techniques to fill in those gaps. At the same time, we have other kinds of data sources. We've got the nursing data, we've got assessment data, which is recorded, certainly in my institution, in a machine readable way. We use a lot of imaging, particularly in my domain of intensive care. There's an awful lot of data there. And it's very difficult for a clinician to actually really synthesize all those bits of data. For a human being to hold more than one or two sort of bivariate kind of things in their head it's just really difficult thing to do, particularly and that's before you start thinking about the dirty, the noisiness and so on, and the informative sampling and so on that's just inherently present in real world data. So that's kind of my day job. I was also involved in introducing the electronic health record in my institution. I'm one of the interim CCIO. I'm an interim CCIO for Cambridge University Hospitals. And so also in my day job is actually trying to persuade clinicians who are using data as part of their care process, but to improve their data quality. First and foremost, I have to say, to improve patient care, but secondly also so that we can secondarily reuse this. And it's a real challenge, and perhaps something we can return to later. My final sort of interest in data, or dirty data, came from, I've been involved in a variety of sort of multinational and local and UK initiatives where I've led on data curation. And really data curation is, as you said, it's a Cinderella subject, in that no one thinks it's terribly important. It's boring. You just assume it'll be fine. And I can assure you from my personal experiences, it is anything but fine. I liken it to an onion, that you take some data and every time you peel off another layer, you find another problem with it. And every time you peel off a layer, it makes you cry a little bit more. It's an enormous problem. It's hugely underestimated when people put in for grants and plan their analyses. And it's only to some extent that we can offset that by trying to design the data properly at source. To some extent, there will always be a requirement for dealing with some of the inhomogeneities and dirtiness in the data. And we published some international guidelines on to try and actually raise the awareness of data curation generally.
That's tremendous. I mean, yeah, I'm sure there are a number of points there that we'll be coming back to later on in the discussion. And if those, as they have with me, have triggered questions and thoughts, I do hope other people in the audience will put their thoughts and questions in the chat as we go. Let's move over to Martin. Martin, again, we're hearing some fantastic real world takes on the challenges. So I'm also very interested in, as well as hearing about the challenges we go, just a little bit of, perhaps some of the solutions that you're, so I don't want to downplay the challenges, but to hear about the challenges, but also some of the solutions that you're bringing to bear on those.
Yeah, thanks, Andrew. So first of all, I'm a general surgeon, so adding on the heterogeneity of data, when Evis said about imaging and Ari told us about the vital parameters being recorded, what I'm most interested in, the procedure of surgery and the huge amount of data we actually produce during the surgery and then most times throw away, basically as much as Ari does it. So that's the reason that I myself and my group, we focus a lot on working with imaging, but on surgical video data, which is surprisingly different for the computer scientists to work on, because if you have a computer scientist working on medical imaging, being CT and MRI, they most certainly fail when working on video. So this is something I've been working on, and actually it's for us as surgeons, it's nice because we can contribute this video and the information it contains to decision making in a, let's say, multi-omics scenario. But at the same time, if we just focus on the video, we can use machine learning, probably not in the sense Mihaela was talking about, but by providing intraoperative assistance. So this is, I think, something I'd like to add to the discussion. It's not only important to have the data, but to know what do you need the data for. So you can collect much of data and you can clean it and it's not dirty anymore and you can do fantastic machine learning on it, but it's important that you choose a clinical decision you want to inform or improve, because, you know, there's one thing where you have all the data available and you just don't have a look at it at the right time. So if we see clinical data as a stream in the treatment pathway and we find the right moment to just inform people, here's something may be wrong, what do you think? People will be smart enough to make the decision, and just machine learning could help to focus their attention. The second thing, and I think this will be most certainly only happen in intensive care, is what Ari already said, that the amount of data and the different kinds of data are so complex. For example, in microbiology, which antibiotic to choose for which patient at which time. I think here you need so much expert knowledge that even in some university hospitals, it's not always around, and certainly not around the clock. So I totally agree with what Evis and Ari said about the importance of different kinds of data and entering data in a structured manner in the first place, instead of trying to extract structure from unstructured text, for example. But what I think is also important, and what I experienced in my research when I did some nice machine learning work and predicting survival or complications after major surgery, my fellow surgeons asked me, what's the use? I do know that the tumor stadium is important for the prognosis, but how can I use this in making clinical decisions? I think this is very important.
You raise some really important questions and points and make some important observations there, Martin. And I'm just going to break the cycle of going around the panel, just because you know, it's almost like you've been eavesdropping on the conversations going on in Mihaela's lab. So I'm just going to ask Mihaela just to comment on a couple of points that you made there. You know, particularly about this. It's not, you have to know the purpose, that the data, you think about the data differently if you know what you're going to use the data for. Mihaela, I don't know if that's something you want to talk about.
Well, I found it unbelievably interesting that both Ari and Martin mentioned that, because to some extent, I think that there is a paradigm shift even for us, the machine learners, that your domain is bringing to us. Because currently, I think if you look at many of the publications in the medical domain, you will see people creating the cohort for the analysis in the medical paper, where they are going to say, we don't have enough of these variables, or we don't have enough patients with these characteristics, or we don't have enough clean data for this purpose. And increasingly, we are building machine learning methods for that, rather than just making the predictions, because maybe the predictions can be done with a Cox survival model and they will be equally good. But where machine learning maybe can help even more is trying really to understand what is valuable data in the context of a particular outcome or a particular decision that needs to be made. And it may be that we do not have enough data for decision one or purpose one, but we do have enough data for another decision. And maybe we can impute in some scenarios and still measure the effect of interest, while in other scenarios, we even don't need to consider that particular data. So this issue of value and value of information for a subgroup of the population in the context of different tasks is very much where machine learning is currently looking at. And we are developing technologies really to address this type of questions. What's valuable data for what question? So it is nice that this is highlighted here as well.
Very good. Thank you, Mihaela. And we're going to come back, I'm sure, to the to this recurrent that we've heard 3 times now about the challenges of data quality, but also of the heterogeneous nature of the data that all of you seem to be dealing with. We think of medicine as being highly specialized and kind of, but in fact, what you're all showing is that even within your specialisms, there is an enormous amount of heterogeneity. Suetonia, you're a familiar face to this audience, but your the areas that you deal with are so relevant, and I think we haven't really heard from you today so much on the data aspects of it. So we'd love to hear from you, because you've really taken the bull by the horns, I think, on some of these data issues. So Suetonia, do explain to the audience your connection with our topic today.
Thank you, Andrew. So I'm a kidney specialist, and my clinical work is working with people on dialysis and transplantation, and particularly kidney transplantation. I think, like Ari said, we are a data-rich area because we've got lots of fancy machines that provide people with treatment over a very long period of time. So we know our patients for often decades, but what we don't have is really good information about what helps survival. So we're, unlike ICU, we're in an area where no matter what trial we do in the area of dialysis, the answer is always negative. Whatever we're trying to do to change someone's survival or help improve their quality of life, unfortunately, the trials are invariably negative. And so we are in an area where I don't think we truly understand what is driving survival and well-being for our patients, because all of the contributing factors to their lives, there are very, very many of them. The disease is incredibly complex. I think, like ICU, the diseases are very, very complex, and we don't actually really understand the diseases. And so that's, I think, where machine learning comes in for us, is actually understanding what are the true drivers of well-being for our patients, and can we modify them? And we're not even at that stage yet. The wonderful thing is that we're data rich, but we're very reliant on the data that are collected, and the data have been collected since the 70s, well before many of these ideas have even come to fruition. And so the data don't include what we now know or think we would like to know. So we're in a data shift mode where we're trying to change the way we collect data, and we make it much more routine. So getting it into the electronic health record, but we're very reliant on older data and older style data, but we're trying to make much more modern decisions. And the data that we tend to miss is around medications, and also what happens to the patient in their actual lives. We capture them when they're on dialysis, but we don't know how mobile they are, we don't know how frail they are, we don't even know where they live, how long it takes them to get to treatments. And I think those are the data that we actually really need to capture. And I think augmenting our existing data sets with other really, really informative data about people's lives has privacy concerns, geolocation concerns, cross-border concerns, and also the potential for reinventing the wheel. We've got a dominant population on dialysis, but we don't capture a large number of other factors. Many of our patients are homeless related to their treatment, or are in dire socioeconomic conditions. They may not be as prominent in our data set. And I really like Mihaela's concept of synthetic data and semi-synthetic data to enhance the presence of many patients in our data set, so that we can actually understand them better, so that we don't recreate biases that we already are using in our standard statistical models.
Thanks, Suetonia. I mean, you raised so many amazing points. I mean, how this idea that, I mean, it's kind of this idea that since, you know, everything you try, nothing works, but you have a sort of a belief underlying all of that, that the answer's in there somewhere. I mean, it maybe not in the data you have today. I mean, it's sort of, could you just give us a little more on that? You know, you said about mobility and frailty and how long to treatments and, you know, what else going on. Is this sort of, I won't say desperation about, well, nothing else has yielded it, so it must lie in there somewhere? Or could you just help us? I think one of the things that also is so important in data is one of the things that Mihaela talked about as the fourth antidote, which is using your expertise. And that's what I think we're hearing here, which is using your clinical expertise to drive the data gathering. Can you just talk a little bit more about that insight?
Yeah, I don't think we've run out of space or room on the runway. And I actually wonder if we've been looking in the wrong direction, to be honest. And that's born of decades of negative trials. But I actually think we don't truly understand what drives people's well-being. And we are only glimpsing the top of the iceberg, I think, when we're thinking about medications. Of course, I'm not a surgeon, and surgeons and intensivists and radiologists have very powerful intervention tools. And obviously transplantation is a transformative intervention. So let's not forget that. But within places where people are very, very sick, I don't know that we're looking in the right places. And I think that machine learning can help us actually understand the importance and the value of non-traditional ways of understanding well-being and measuring them and intervening. Yeah,
it's a really, as I say, it's really powerful reinforcement of Mihaela's fourth antidote. I think we've just heard it in action in your answer. Doug, I hope you're still with us. We can't see you, but I hope we can still hear you. Doug, are you there?
Oh yeah. Hey.
We can hear you and see you. Perfect combination.
Yes, amazing. I've been listening carefully. I finally figured out how to get the speaker working on my phone. So yeah, I'm an internist at, I should introduce myself, right? I'm actually at about to go to a conference in Irvine on data. So it's very apropos, and I'm standing outside the building, but I direct the informatics program at the Translation Institute at UCLA. We provide the EHR data for studies that need it, and we help them interpret the data. So we do about 150 data sets a year, my team does, and so data quality is really, really important. And we've seen really big problems with data quality that we sometimes catch. One that comes to mind was we were providing data on the whole EHR to for our core discovery, and we noticed that there was a lab was mapped as a medication instead, and we saw there were like 2 million orders for toe fungus medication. And so we knew that, there's got to be a problem there, and we finally figured that out, what the problem was. But, that's just one example. And, it's frequent that we really have to get into the nitty gritty of a particular data set and look at, what might really be going on. And, we see mapping problems all the time. And, we find errors, a lot of errors get introduced. I was, we help people interpret the data a lot, as I said, and I would just underscore it's so important to look at what they need, really. What's the hypothesis that an investigator is really trying to get at in order to, really make sure we get the data that they really need for that. So I'm sorry, there's background noise. So we spend a good deal of effort on that, and so I think machine learning, we're not doing a lot with machine learning yet. We are we have been using some natural language processing transformer models to do extract concepts from pathology reports primarily, and we had some very good results with that. But I think we're just at the start, really, of applying machine learning in practice. So, machine learning for data quality is going to be very, very important, I think.
That's tremendous, Doug. Really interesting insight. I'm wondering, though, from you but the rest of the panel, maybe people can just put up their physical hands, since I can see you all. Just who has? I'm interested also in just your experiences, perhaps feeding off what others have said as well, of where you've used machine learning techniques to either compensate for or solve the data issues. Ari.
Yeah, so I mean, I think one area to make Suetonia feel a little bit better, intensive care is another area that precisely 0 trials have ever shown any effect. So don't worry, you're not alone. And again, I think part of it is because of heterogeneity. So take 1,000 sepsis patients. Well, what sepsis, what bacteria, and where, when, what kind of patient, what genetics behind the patient, but frankly, how much fluid did they get? What did you do to, so many different things to say about so it's completely heterogeneous. And in my area, a lot of my patients are traumatic brain injury patients. Now we categorize them as TBI patients, acute, mild, moderate, and severe TBI on their level of consciousness. Now that's like me, if I was to go to an oncologist and say, I'd like you to categorize cancer as mild, moderate, and severe, they just laugh at me. I mean, it's a laughable way of describing something that's so complex. And yet we have a huge amount of data, but we can't synthesize that into a better endotype of the disease. So certainly one area that we've been looking at quite hard in traumatic brain injury, which again it's a relatively rare, well, globally common but relatively rare, is to use clustering methods to both in temporal clustering and at fixed points in time to try and understand, to try and come up with better phenotypes or subphenotypes, or ideally suggestive endotypes that we can then move forwards to do more targeted therapies and targeted trials. So we enrich our population, hopefully, for particular subclassifications of disease that are more biologically meaningful and then yield a positive result.
Can you give an example, Ari, of where you've identified, or the machine, I'm sorry, has identified a cluster that you've realized is actually a biologically significant cluster that can be treated in a particular way?
Yeah, and again, I think this sort of speaks to one of the points that Mihaela made, that another thing about machine learning, about traumatic brain injury, is that we have some really strong predictors of outcome. So your fancy machine learning algorithm isn't going to do better at predicting outcome than logistic regression, or not really very much better. But what it is really good at is taking some really complicated data and clustering it in a robust sort of way, which in itself is not an easy thing to do. So we've published quite recently looking at endotypes of TBI, and we found some interesting candidate endotypes which show particular biomarkers for physiological stress. And then when you look at parameters that outcomes in those particular endotypes, they differ. So all the things you think would be the same kinds of patients, actually these are differentiated by biomarkers of physiological stress. Now, the thing we haven't done yet, but there are areas such as ARDS where relatively simple latent classification algorithms have found endotypes, have gone forwards and actually tried and tested different therapies in those different endotypes and found differences. So if you treat endotype 1 differently to endotype 2, you can get better outcomes. And we haven't done that yet. And that's the final thing that you always have to do with these things, to actually prove that they're useful. But it's very much an interesting hypothesis generating sort of space.
So, that's an example of dealing with the heterogeneity with by identifying clusters that potentially humans wouldn't have been able to pick up given the complexity. I wonder, Suetonia, please.
Yes, I think the other way around to think about it might be that machine learning can help us understand, because Ari, I think your wonderful insight is that we actually don't know what disease is and what we're actually talking about. And actually, I think machine learning can take us back to the beginning and refocus us on how we think about what disease actually is and who's got what, because then that can help us untease how we can help people differently depending on the disease. So I think phenotypically we cluster people in our heads and in an expert way, but we probably actually have to go back right to the beginning and question how we think about phenotypes and how we think about disease. One other way that machine learning can actually help us with this is Ari's lovely insight around average treatment effects. So we go into a trial with 10, 20,000 people, and we hope that we're going to see that all of them, every single one of them, have the same treatment effect. And then we get despondent when they don't, which is not really very surprising. But if we can understand how individuals respond to treatment using machine learning, then we can probably go back almost and then look at what their phenotype is, or understand more about them from the other end. So we understand what the treatment, who the treatment helps, and does that help us untease who they are and why it helps. So we come back a little bit to mechanism, plausibility, and biological processes that help us re-understand diseases. I actually think it's that profound.
It is. I mean, that does sound like a very profound shift, a very important one. Martin, and then I know Mihaela has something she wants to say. Martin first.
Yeah, I'd like to add something on the differentiating disease phenotypes. I think we also maybe should focus a little bit on the treatment phenotypes, I'd like to say, because we have a good manufacturing practice for the new drugs that are being developed, and intensive care, as all of you know, I prescribe an antibiotic 3 times a day at 8, 16, and midnight, and it's given at 7 and 18. And I think even more for my profession, it's what we increasingly do, that in randomized controlled trials, we have quality standards for surgeons performing in both arms of the trial, because treatment itself is can be clustered in different types of treatment. And I think this is something we must not forget. And I think it's not anything human can do, because a human doctor describes a therapy and does it the best way. But machine learning may detect from automatically derived data what phenotypes we have here.
It's great. It's a great insight, Martin. Mihaela.
First, I want to show a lot of excitement as a person being on the other side, because I think that these problems that you are highlighting here, both, let's say, outcome-driven phenotyping, so it's not only clustering and endotypes, but also how a particular, because 2 different types of patients may look very different, but yet have the same outcomes, and vice versa. So with respect to outcome, this is a problem that you will not believe it, but machine learning community has not looked at it. And a few years back, it is a clinician like you who gave this problem to us, and we started to develop this framework of outcome-driven phenotyping. And this is now quite a booming area in machine learning, but it came from one of your side. And I was very excited that Suetonia and Martin, that now you give us a new problem of this treatment, how to cluster treatment and treatment outcome, treatment response driven clustering. So mission accepted. I just wanted to say that you do not realize how much these conversations are also triggering ideas for us and the machine learning community. And I'm quite keen to bring these challenges that you pose to us to the machine learning community. For some things, methods exist, but we can do a lot better. So please keep providing this type of challenges to us.
Very much so. Evis, do you have a comment you want to add?
I have 2 quick comments. One, something that really bothers me is the amount of failed clinical trials we have, especially in oncology. I mean, and everywhere maybe, but I just don't think the trials I'm aware. And not just pharma trials on a new drug, it's just really those trials funded by public funding, types of MRC, and and I just think it's one of the reasons for sure in my field, I know in ovarian cancer, is because what we're using to measure response and to categorize response, it's wrong, basically, So let's say resistance measurement, on a disease like ovarian cancer, it makes absolute no sense because the disease is not linear, you just can't measure A to B. So, but there are many examples of this. So I wonder, how would machine learning, if you have to go back and get the data for those trials, would you be able maybe to sort of untangle that and really offer a solution to better reclassification of responders, not responders. This is one. And the second one is the frequency of following up patients. And this may be a cancer patient, maybe non-cancer patients. It's so empirical. I mean, I've worked in 3 different countries with the same disease, the same parameters. The follow-up doing a CT or an MRI is absolutely different. It's a mess. And I think you'd say that, well, sometimes if you use MRI, there is no radiation, that and that. Actually, it still costs an anxiety. Anxiety. So I think, how do we redesign that sort of follow-up, or, and then, yes, you take into account the stage, the outcome, that and that, but do you take into account patient's immune status? Do you take into account their sort of home devices? I don't know. I mean, I just think it's, anyway.
Well, no, it's both your points actually, but are very close to Mihaela's heart, I know. So I'm going to hand over to Mihaela to give your response.
So first, I want to highlight some of that, not for the case of cancer first, but for the case of ICU. So together with Ari, we developed a package actually, which is looking at ICU and hospital data, where we call that clairvoyance. Ari's name, he baptized this package as clairvoyance. But as part of that package, this is kind of exactly what we try to do, because even in ICU, and in hospital, there is this question of how often should I measure something, what type of variables, and for what purpose, and at what frequency. And if I measure one thing, what is the importance of measuring another thing at another moment in time? So in the context of ICU, Ari and I have done a piece of work that is publicly available, and code is available for it. So if you are interested in that, please reach out and take a look. But what's even more exciting, because much less I think has been done, is what Evis is saying now, which is what modality I should give when and how. And I myself moved in 4 different countries over my lifespan, and I'm at very high risk of breast cancer. And this question of how often should I be monitored and what type of technology has been very much to my heart as a patient, as a patient at risk. And I think that the technology exists in terms of machine learning to estimate what would be the value for a class of patients at looking at these different types of technologies. So what I'd like to say is, Evis, let's join forces to try to see if we can work out some of this and then bring an exemplar to this group again and try to hopefully shed some light on it.
I can see we're going to run out of time today, and that's a good thing, that there's so much we could do. But Ari, you have an insight you want to share?
Yeah, I was just going to talk. I was going to also come back to Evis's point, which I thought, points, in fact, which I think are really interesting. So I'm also now going to stick the knife into randomized controlled trials a little bit more. And I'm not saying that they're not the gold standard, because clearly they are the gold standard. But I just want to point out that the gold standard isn't, is a little bit tarnished, really. Even randomized controlled trials don't, both philosophically and mathematically, don't guarantee causality. Okay, and on top of that, the generalizability of randomized control trials, because when you do make one that actually works, it's because you've restricted your inclusion criteria so hard that actually at the end of it, well, does it really apply to the patients that you're in fact looking at? So I'm not saying we shouldn't be doing randomized control trials, and there are some areas, of course, where randomized control trials work very well. But if we accept that the gold standard isn't that shiny necessarily, then actually there are lots of other methods of getting towards causality, because that's essentially what we're trying to do here. And so, if randomized control trials just aren't going to work, and in intensive care, I've got a countless list of how many that have just fail all the time. There'll be another 10 this year, I'm sure. Then actually maybe we should just abandon this. Maybe this isn't the right way of doing things, and we should use other data-driven methodologies for for assessing causality. And the second thing is just in terms of what the right treatment or the right follow-up or the right imaging is. I'd just like to make that even more complicated, which is it has to work within the system of care as well. And this comes a little bit towards Martin's point too, that it's about exactly what you do and whether you do it right. So even once you've decided what the thing that you should be doing, do you actually do it? Because there's what we should do, what we think we should do, what we think we do, and what we actually do. And those things are not necessarily all the same thing, and they can have huge impact in some domains to outcome.
Harry, those are great points. I feel like we're about 10% through the conversation that we should be having, and unfortunately we're out of time. I know there were a couple of questions we didn't get to on the chat. I saw that Mihaela gave a great answer to one of them on the chat. We'll do our best to get to the others in a offline fashion, but I didn't want to break the flow of the conversation that was going on in front of us all. I want to thank our panel, which has just been an amazing discussion. I'm fizzing with thoughts and reflections, and so that's been amazing. I hope it's been useful for our panelists, but more importantly, I hope it's also been as useful as it has for me, for the audience. Doug is our first kind of on location panelist that we've had, and Doug, I really appreciate you taking time in a busy day to join us, but also all our other panelists. To everyone else, thank you very much, and a lot of interest and demand for more on clinical trials. So maybe perhaps Mihaela will give some thought to whether or not clinical trials would be a great subject for a Revolutionizing Healthcare session.
I would like to highlight the fact that we had 2 sessions on clinical trials that I'd like the panelists and everyone to take a look also. So last year in the summer, we have 2 sessions that were very well attended on clinical trials. So please take a look. Some of the things you raised were discussed there, but a lot not. But still, please take a look.
And we'll send around the links to those when we send around the links to this, when the recording of this session. So thanks very much, everyone, for coming along. Really appreciate your time, and we look forward to seeing you again next time. Bye-bye.
Thanks so much. Bye-bye.